Welcome back to Advent and another interesting episode of Dr. Mohanty's Masterclass. Today we are going to discuss a topic that has gained tremendous importance in the pharmaceutical and chemical industries in recent years, that is nitrosamines. These compounds, though small in structure, have raised big concerns worldwide due to their potential health risks. What are nitrosamines? Nitrosamines are a class of chemical compounds characterized by the presence of a nitroso group attached to an amine. What makes them a concern is their biological activity. Many nitrosamines are not only classified as probable human carcinogens but are also mutagenic, meaning they can directly alter DNA and increase the risk of cancer. This dual nature makes them particularly dangerous even at extremely low levels. For example, NDMA, N-nitrosodimethylamine, and NDEA, N-nitrosodiethylamine, are two well-known nitrosamines. Both have been detected in certain pharmaceutical products in recent years and are strongly linked to cancer risk. Another example is NMBA, that is N-nitroso, N-methyl 4-aminobutyric acid, which triggered recurrence in some of the antihypertensive drugs. How they are formed? They are not usually produced intentionally, but rather form as byproducts or impurities during chemical reactions, processing, or even storage. They are typically formed when nitrosating agents such as nitrites react with secondary or tertiary amines under favorable conditions. Situations like acidic environments or high temperatures make this reaction more likely. In the pharmaceutical context, there are several possible sources of nitrosamine formation. During API synthesis, nitrites such as sodium nitrite or other nitrite impurities present in solvents can react with secondary or tertiary amines used in the process. Cross-contamination. Residual sodium nitrite in solvents or process streams may persist due to inefficient cleaning of manufacturing equipment, leading to unintended nitrosamine formation. And third, from raw materials and excipients. Poorly controlled raw materials, starting materials, excipients, or reagents obtained from inadequately qualified suppliers can introduce nitrite impurities, increasing the risk of nitrosamine generation. How they have evolved over the years. Let me come to that. The nitrosamine story in pharmaceuticals really began in July 2018, when the first detection of NDMA, that is N-nitrosodimethylamine, in valsartan, a commonly used antihypertensive drug, shocked the industry. This discovery triggered immediate drug recalls in the US, Europe, and several other countries. By late 2018 and early 2019, other nitrosamines such as NDEA, as I told before, N-nitrosodiethylamine, and NMBA, N-nitroso N-methyl 4-aminobutyric acid, were identified in additional batches of valsartan and even in other certain drugs like losartan and irbesartan. This showed that the problem was not isolated but systemic, linked to raw materials, processes, and solvents. By 2019, the European Medicines Agency, EMA, and the US FDA had both issued urgent guidance asking all pharmaceutical manufacturers to review their processes, assess risks of nitrosamine formation, and test their products accordingly. This marked the beginning of global regulatory action. The scope widened further in 2020 when nitrosamines were detected in ranitidine, a widely used anti-ulcer drug. This led to large-scale global recalls and raised the concern that even finished formulations could generate nitrosamines during storage. Finally, in May 2020, the EMA and other regulatory agencies published consolidated guidance documents defining acceptable intake limits, testing requirements, and risk assessment strategies for nitrosamines across all drug classes. Since then, more and more nitrosamines are being identified as contaminants. And as on today, they had transformed from an unexpected impurity into a worldwide regulatory priority, reshaping how the entire industry looks at drug safety. What are exactly the concerns? Obviously, nitrosamines have emerged as a significant concern, posing various challenges and impacting the industry across multiple fronts. Besides posing potential health risks, their discovery has led to a series of drug recalls worldwide, as I told. Some widely used medicines, such as certain antihypertensives like certain series, were withdrawn from the market, creating a huge impact on both patients and the healthcare system. These recalls didn't just stop at the drug shelves, they triggered supply chain disruptions. Manufacturers, raw material suppliers, and distributors all had to respond quickly, leading to shortages and delays in availability of critical medicines. And finally, there is the issue of economic losses. Companies not only faced direct cost of product recalls, but also regulatory penalties, litigation risks, and loss of reputation. Authorities like the US FDA, EMA, CDSCO, that is Central Drugs Standard Control Organization, under Directorate General of Health Services, Ministry of Health and Family Welfare, Government of India, have issued detailed guidelines for the identification, control, and permissible limits of nitrosamines in drug products. The limits are extremely low, often in the parts per billion range, reflecting the seriousness of the risk. This has pushed pharmaceutical manufacturers worldwide to conduct comprehensive risk assessments. This means evaluating every step of the manufacturing process, from raw materials and solvents to excipients, reagents, and even packaging materials. Along with risk assessment, they must implement robust analytical testing methods such as LCMS, MS, or GCMS to reliably detect and quantify these impurities at ultra trace levels. Nitrosamine impurities do not just pose a regulatory compliance issue, they ripple into clinical timelines, study designs, and product strategy. The result? Delayed launches, higher costs, and added clinical complexities. How they're detected, how they're analyzed, all these nitrosamines. Detecting and analyzing nitrosamines at such an ultra trace level is a huge analytical challenge and requires sophisticated analytical techniques. Commonly used methods include gas chromatography mass spectrometry, which is highly effective for separating and identifying volatile nitrosamines, liquid chromatography mass spectrometry LCMS, which is suitable for non-volatile nitrosamines and provides high sensitivity and specificity. Third, high performance liquid chromatography or HPLC, which can be used in combination with other detectors for the analysis of nitrosamines. Ion chromatography, the fourth one, useful for detecting nitrite or nitrate ions, which are precursors to nitrosamine formation. And this requires not only high quality solvents and reagents, but also rigorous control of background contamination, because even in minute impurities can interfere with results. What we are doing in Advent, what I gave just now was a brief introduction about nitrosamines, their genesis, evolution, and detection methods. We at Advent have been able to develop and service many commonly required nitrosamines as working standards to our esteemed customers for their quantitative determination in desired drug compounds. Here are some of the representative examples with structures. We have developed more than 50 of them so far, and most of them are in ready stock, available with high purity. Customized packs are also available from milligram to gram scale. Certificate of analysis is provided along with all characterization data such as mass, IR, proton NMR, and carbon 13 NMR, etc. Please get in touch with our sales colleagues or channel partners for your requirements. To summarize, nitrosamines are unintended but highly significant impurities that demand our full attention. From drug manufacturers to raw material suppliers, everyone is now required to adopt safer processes, better quality controls, and cleaner solvents. While this has created challenges, it has also opened the door for innovation in chemical manufacturing and purification. The awareness and regulations surrounding them have reshaped how we think about chemical safety and drug quality. Thank you for joining this introductory session on nitrosamines. In the next session, we'll discuss about NDSRIs, that is nitrosamine drug substance related impurities. I look forward to seeing you in the next masterclass. Till then, stay healthy, stay curious, and explore the wonderful worlds of chemistry. Thank you very much.